Hello, I'm Albert, an attending in hematopathology, and I'll be doing an overview of the WHO hematopoietic classification. First, I'll be covering the methodologies that we use to diagnose things in hematopathology, and then a little bit about the overall structure and epidemiology of the classification, followed by a basic starter set of diagnoses in the major categories, myeloid, B, and T. Broadly speaking, we're trying to identify tumors and place them into one of these roughly 230 entities, which are implicitly predictions about clinical behavior and response or non-response to therapies. And each entity has a few paragraphs to several pages listing clinical presentation, morphology, immunophenotype, and so on. And in this slide in red, I've highlighted the things that have either changed criteria or are new entities since the last edition. Our general approach is to start with the clinical behavior and history, why we got the specimen, what the patients had before, and then look at it under the microscope cytologically and or histologically. Frequently, we'll evaluate protein expression by immunohistochemistry and or flow cytometry, and at this point, we'll typically sign out a diagnosis or a differential diagnosis. A day to a couple weeks later, we'll get the genetics, and then we can put the entire package together and sign out a final diagnosis. When we're integrating these methodologies together, we have to put the information from them into a particular diagnostic context. And so different methodologies take precedence over others depending upon the differential diagnosis that we're considering and the specific criteria in the WHO that we have to hit. One of the more complicated things about hematopathology is the level of detail in which we assess combinations of protein expression. So, for example, here we have a case of classic Hodgkin lymphoma, which has this large Reed-Sternberg cell, the ally cell, and in immunohistochemistry, brown is positive and light blue is negative. So this Reed-Sternberg cell expresses CD30 in a membrane and Golgi pattern. It co-expresses CD15, and it has this dim PAX5, so you can see that there's a faint brown in the nucleus there. And so when we put that whole thing together, we end up with a diagnosis of classic Hodgkin lymphoma. But not infrequently, we don't have a perfect pattern like this. Often, for example, CD15 is negative or PAX5 is negative, and then we have to order additional stains to confirm the diagnosis. And so at the end of the day, it's fairly common to end up with 10 or 15 stains. With flow cytometry, we're getting a lot of the same information, but quantitatively and at the single cell level. So we have cells in liquid suspension that we run individually through the instrument, and we shoot lasers through them to determine their characteristics. For example, here we have a neutrophil, and when we shoot a laser through it, these internal granules will refract and reflect some of the light perpendicularly, and this light will be detected by the side scatter detector. And so each individual neutrophil, because it has this high side scatter signal, gets plotted as an individual dot in the high side scatter region of this plot. Whereas a lymphocyte doesn't have as many of these internal granules, and so the laser will just pass through. It won't have as much of this side scatter signal, and so each individual lymphocyte gets plotted as an individual dot in the low side scatter region of this plot. Similarly, other populations have characteristic regions in the side scatter versus 45 plot, which we then use for downstream analysis. An important population is these blasts, which is a catch-all term for immature progenitors, which normally go on to differentiate into neutrophils, monocytes, lymphocytes, and red cells. Using the side scatter 45 plot, we can then gate or select out specific cell populations, and if we stain all the cells with fluorescent antibodies, we can assess their patterns of protein expression. So here, for example, we've stained all the cells with fluorescent antibody against CD38 or anti-CD38, as well as fluorescent antibody against CD5 or anti-CD5. As we go upwards along the y-axis, we have more reactivity or binding to the CD38 antibody, which we infer to mean there's more expression of the CD38 protein on the cell. Whereas if we go rightwards along the x-axis, we get more binding of the CD5 antibody, and we would infer to mean more expression of the CD5 protein. When we gate on the monocytes, that means that we've selected out this particular region, and each one of these individual dots is now represented as an individual dot in this plot. And we say that the monocytes in this case are expressing CD38 but not CD5. They express CD4 but not CD8, and they lack both CD10 and CD20. Now instead, if we gate on the lymphocyte region here, each one of these individual dots is now represented in this plot. Even though we're looking at the same marker, CD38 versus CD5, CD38 versus CD5, now we're looking at a different set of cells that have different patterns of antigen expression. And whereas in the monocytes, we mainly had one cell population, in the lymphocytes, we have multiple cell populations, including these likely T cells that are subsetted into the 8 positive and the 4 positive subsets. And from prior experience, we also know that these brightly 38 positive cells correspond to these 2 subsets of hematogones in this kind of stair-step pattern on the 1020 plot. So when you're looking at these plots, you have to know not only what are the normal patterns of antigen expression, but also what are the abnormal patterns that are characteristic of various diseases. So with your typical heme path case, you have your morphologic impression, which you then have to further interrogate by immunophenotyping, either immunohistochemistry or flow cytometry or both. And with flow cytometry, you have this additional complication where we have to split the sample up into multiple tubes, where each tube has a different combination of antibodies in it. And that means that we end up having to do this 15-dimensional Sudoku problem trying to line up cell populations across plots, across tubes, and then back correlate that with what we saw under the microscope. And integrating both of these things together is what gets us to a diagnosis. Okay, so now on to the actual structure and organization of the WHO. This is a picture of Nevada and vernal fall in Yosemite, which is just a nice picture. Hematopoietic tumors account for about 6 to 9% of cancer cases and deaths, and they tend to occur in older individuals, 60 plus. Although acute leukemias are the most common cancer of younger children, and lymphomas are among the most common in adolescents. And most of them occur pretty comparably in males and females. The way that the WHO classification is structured very much mirrors our understanding of the hematopoietic tree, where we start with this multipotent hematopoietic stem cell that can differentiate along a myeloid arm or a lymphoid arm. The myeloid arm produces megakaryocytes, which spawn platelets for clotting, red cells which carry oxygen, and then various granulocytes for fighting infection as part of the innate immune response. The lymphoid arm produces our adaptive immune cells, B cells and T cells, which undergo VDJ recombination for antigen receptor diversity, as well as NK cells, which are considered part of the innate immune response. And then our B cells can further differentiate into longer-lived plasma cells, which act as antibody factories. All hematopoietic tumors are thought to originate from some normal counterpart along this tree. So they're primarily organized by lineage, either myeloid, which I've shaded blue, B shaded gray, or T in green. But at these early stages, they can be ambiguous, either uncommitted or having characteristics of multiple lineages. So putting these things together, most of the entities in the WHO are organized like this by presumed cell of origin. There is a category of tumors associated with immune deficiency and dysregulation, where people who are immunosuppressed are more susceptible to oncogenic viruses such as Epstein-Barr virus, Kaposi sarcoma-associated herpes virus, and human herpes virus 8. And these viruses can transform normal B and T cells into lymphomas, which look just like lymphomas in non-immunosuppressed people, except these ones are positive for one or more of these viruses, and they occur in the context of immunosuppression. This category also includes some proliferations that are not overtly malignant. The last two categories I'm not really going to talk about in this presentation because they're on the rarer side of things. The first being dendritic cell and histiocytic neoplasms, most of which are thought to derive from monocytes and macrophages. And then there are stromal-derived neoplasms of lymphoid tissues, which are not hematopoietic in origin, more they derive from the stromal cells that support hematopoietic tissues. If you break down the major hematopoietic neoplasms by incidence in a pie chart, what you see is that about 1/3 are myeloid neoplasms, from here systemic mastocytosis to here AML, and about 60% are B and plasma cell things, from here plasma cell myeloma all the way around to here B lymphoblastic leukemia. And only about 5% are T-cell neoplasms, from here mycosis fungoides around to here T lymphoblastic leukemia. When it comes to identifying cells and tumors immunophenotypically, this list covers most of the markers we use by flow cytometry and immunohistochemistry, as well as the cell populations that normally express them. We use CD45 as a general pan-hematopoietic marker, mainly if we're doing a small round blue cell tumor workup, but there are a lot of exceptions. It's useful to know that in PowerPath, when you order the CD45RB, I think it's actually a pan-CD45. It's a mixture of two clones, 2B11 and PD7/26. PD7/26 is RB specific, but 2B11 is a pan-45 clone, so I think when you order 45RB, you're actually getting a pan-45, which I think is more useful. For defining lineage, the most important markers are CD3 for T cell, CD19 for B cell, and MPO for myeloid. But for myeloid cells, it really depends on what subbranch of differentiation you're looking at. There's one set of markers for erythroid, one set for neutrophilic granulocytes, one set for monocytes, and so on. So it's really hard to just pick one or two. In Western countries, the most common tumors we see are B-cell origins, so you'll also see CD20, which is good for more mature B cells, PAX5, which is kind of like a nuclear version of CD19, and CD138, which is fairly specific for plasma cells. In the hematopoietic system, CD56 is used as a marker for NK cells, but it's neither sensitive nor specific, so in general, we just don't have a great marker for NK cells. And when we're looking for acute leukemias or kind of immature primitive neoplasms that behave a little more aggressively, CD34 is a fairly good marker across all lineages. CD117 is more myeloid oriented, and TDT is more lymphoid oriented. Getting into the actual entities, we're going to start with the myeloid things. This is a very nice case of acute promyelocytic leukemia, or APL, which is important to recognize on call because these patients have a propensity to go into DIC and suffer from strokes unless they're given ATRA, or all-trans retinoic acid. The classic description is your cell with a butterfly-shaped nucleus, like this or like this, with some granulation in the cytoplasm. But you can see that there are varied morphologies, including some that look like regular nondescript blasts from any old regular leukemia, whereas some are very granulated without really obvious nuclear lobation. And I find many of the cells will end up having more of a lumpy, bumpy nucleus rather than a really obvious bilobation. If you're uncertain when you're on call, just kind of keep looking at multiple cells, get an overall impression rather than trying to rely on an individual cell. And you can always call a fellow or call your attending. Myeloid neoplasms are this portion of the hematopoietic tree and this portion of the incidence pie chart. The way that we break down myeloid neoplasms is starting with the myeloproliferative neoplasms, which are characterized by increases in the CBC counts of RBCs or neutrophils and or platelets, but without morphologically abnormal cells or dysplasia. That contrasts with the myelodysplastic neoplasms, where you have decreases in the CBC counts of red cells, neutrophils, and or platelets, and also accompanied by morphologic dysplasia. In between, we have this category of myelodysplastic myeloproliferative neoplasms that have features of both. They have increases in the CBC counts of neutrophils and or monocytes and or platelets, which are a myeloproliferative feature, but they also have this morphologic dysplasia, which is a myelodysplastic feature. Next, we have acute myeloid leukemia, which is primarily a proliferation of immature progenitor cells, or blasts, and tends to behave more aggressively than the other myeloid neoplasms. It can occur de novo, or without a prior history of a myeloid neoplasm, or it can occur as a progression or evolution from one of these other myeloid neoplasms. Traditionally, having at least 20% of nucleated cells in the marrow or blood being blasts would get you to a diagnosis of AML. But there are certain genetic alterations which allow you to call AML below that 20% threshold. And by flow cytometry and immunohistochemistry, CD34 is currently our best marker. But in many leukemias, it's only partially expressed or it's not expressed at all, particularly those with monocytic differentiation or acute promyelocytic leukemia, APL. So not infrequently, the diagnosis of AML has to be made, or is most easily made, with just 2 eyeballs and a brain. And last but not least, we have systemic mastocytosis, which is a proliferation of mast cells. All of the myeloid neoplasms are thought to involve mutations in immature progenitors, which look roughly something like this under the microscope. So morphologically, we just group them all into this catch-all of blasts. When they acquire a JAK2 mutation, which causes overproliferation of red cells, this results in a disease called polycythemia vera, or PV. And these patients primarily have increased RBC counts and increased hemoglobins. When progenitors acquire a JAK2 CALR or MPL mutation, which results in excess proliferations of platelets, the result is a disease called essential thrombocythemia, or ET. The T922 BCR-ABL1 translocation can cause a proliferation primarily in the granulocytic arm, resulting in chronic myeloid leukemia. And in these patients, the primary increase is in neutrophils, or neutrophilia, but typically these patients also have a basophilia and eosinophilia and increases in myelocytes. Thrombocytosis is also quite frequent. Our last major myeloproliferative neoplasm is primary myelofibrosis, which typically shows neutrophilia and or thrombocytosis resulting from JAK2 calreticulin or MPL mutations. Mutations which give us proliferations of monocytes, most frequently these mutations are in ASXL1 and TET2, result in chronic myelomonocytic leukemia, or CMML. And this disease differs from the myeloproliferative neoplasms in that we frequently see morphologic dysplasia. So the dysplasia makes CMML the most prototypical and the most common MDS-MPN, or myelodysplastic myeloproliferative neoplasm. Also in the same category is a disease now called MDS-MPN with neutrophilia, and this primarily shows neutrophilia, but the neutrophils are dysplastic. And this disease used to be called atypical CML, but quote-unquote atypical CML had nothing to do with CML, and that just confused people, so they renamed it. Another MDS-MPN not shown here is myelodysplastic neoplasm with SF3B1 and thrombocytosis, which typically shows anemia and thrombocytosis with an SF3B1 mutation. So in red, we have the 4 main MPNs, and then in blue, we have our MDS-MPNs. c-KIT D816V mutations, which cause neoplastic proliferations of mast cells, result in systemic mastocytosis. And one of the interesting things about this disease is that it is frequently associated with proliferations in other arms of hematopoiesis, or AHNMD, associated hematologic non-mast cell disease. And the most common AHNMD is actually CMML. In myelodysplastic neoplasms, mutations in the hematopoietic progenitors dysregulate the differentiating progeny, causing apoptosis in the marrow, and this results in decreased counts on the CBC, anemia, thrombocytopenia, and neutropenia. And in about a third of these patients, these blasts begin to proliferate uncontrollably, and they go on to become AML, or acute myeloid leukemia. The WHO has a very nice table summarizing the morphologic features of the myeloproliferative neoplasms other than CML because they can mimic one another morphologically and clinically. For example, if somebody has PV but they've proliferated through their iron, they might be iron deficient and the erythrocytosis might not be as obvious. Prefibrotic primary myelofibrosis can mimic essential thrombocythemia in the early stages if they just both present as thrombocytosis. So often enough, we don't top line these as just this is what the diagnosis is. We'll frequently give a differential diagnosis and then describe the morphologic features supporting each thing in the differential. So PV morphologically typically has panmyelosis, which means increases in all three lineages, granulocytes, erythroids, and megakaryocytes. Whereas ET is typically only increases in the megakaryocytes, and then primary myelofibrosis is typically increases in the granulocytes and megakaryocytes. PV and ET megakaryocytes tend to have this kind of staghorn nuclear morphology. So the nuclei have these kind of deep lobulations, whereas PMF megakaryocytes tend to be hypolobulated, where the nuclei are kind of aggregated together, and so they have a more quote-unquote cloud-like morphology. PMF megakaryocytes also tend to form these tight clusters, whereas ET megakaryocytes tend to be more loosely grouped and kind of scattered. The myeloproliferative neoplasms have relatively well-defined genetics that I've discussed before, with the T922 translocation in CML, JAK2 mutations in PV, JAK2 calreticulin and MIPL mutations in ET and PMF, KIT mutations in systemic mastocytosis, and CSF3R mutations in chronic neutrophilic leukemia, or CNL, which is one of the less common myeloproliferative neoplasms, which has increases in neutrophils but without morphologic dysplasia. With myelodysplastic neoplasms, the WHO 5th edition designates a framework where a few genetic abnormalities are set aside, but with a lot of fine print. And outside of these few genetic abnormalities, it becomes MDS morphologically defined. MDS with low blasts and isolated 5q deletion is associated with favorable prognosis and possible response to lenalidomide. But this category excludes cases with more than one additional karyotypic abnormality, cases with monosomy 7 or 7q deletion, and cases with biallelic TP53 inactivation. MDS with low blasts and SF3B1 mutations seem to form a distinct group with favorable prognosis, and typically you see ring sideroblasts in these cases. But this category excludes cases with 5q deletion, monosomy 7, complex karyotype, and biallelic TP53 inactivation. MDS with biallelic TP53 inactivation, unlike the other two groups, is associated with unfavorable prognosis and frequently seen with multiple karyotypic abnormalities. And so this class supersedes MDS with 5q and MDS with SF3B1. For the most part, MDS morphologically defined is subdivided by blast count, where you have MDS with low blasts and MDS with increased blasts, and MDS with increased blasts is further subdivided into MDS IB1 and MDS IB2. And you have these blast cutoffs which are different between peripheral blood and marrow. MDS hypoplastic seems to involve T-cell mediated attack on progenitor cells, and MDS with fibrosis seems to have cytokine expression causing myelofibrosis. The subclassification of acute myeloid leukemia follows a fairly complicated algorithm, but one way to break it down is to start with the blast percentage. And if you don't have at least 20% blasts, you need to look for one of these genetic abnormalities. If you don't have one of these, you can't call it AML below 20% blasts. If you do have one of these, you might be able to call it AML with defining genetic abnormalities. The traditional ones that allowed you to call AML below 20% are T1517 PML-ROR alpha in acute promyelocytic leukemia, Th21 RUNX1 RUNX1T1, and inversion 16 or T1616 CBFB MYH11. And these three are associated with favorable prognosis. T922 BCR-ABL1 and CEBP alpha are part of the category of AML with defining genetic abnormalities, but unlike the others, they retain the 20% cutoff. So if you have one of these two but less than 20% blasts, you can't call it AML. But even with the rest of these, the WHO provides the caveat, quote, elimination of the blast cutoff requires correlation between morphologic findings and molecular and genetic studies to ensure that the defining abnormality is driving the disease pathology. Now, if you do have greater than 20% blasts, you need to look for myeloid phenotype through flow cytometry, immunohistochemistry, or looking for Auer rods. If you don't have myeloid phenotype, then you can't call it AML. If you do have myeloid phenotype, you need to look for a history of Down syndrome. And in the context of Down syndrome, greater than 20% myeloid blasts is either myeloid leukemia of Down syndrome or transient abnormal myelopoiesis of Down syndrome. TAM of Down syndrome is a proliferation primarily in newborns, which looks just like AML, except if you wait a while, it goes away. The distinction between MLDS and TAM needs to be made clinically. And both of these fall under this overall umbrella of myeloid neoplasms associated with germline predisposition, which we'll get into a little more later. Next, you need to look for one of these relatively rare chromosomal rearrangements of these genes, which characterize myeloid lymphoid neoplasms with tyrosine kinase gene fusions, which we'll discuss a bit more later on. Now, we again look for these recurrent genetic abnormalities, but because now we have greater than 20% blasts, we are going to include T922 BCR-ABL and CBP alpha. And if present, it falls into AML with defining genetic abnormalities. Finally, we need to look for a history of a myelodysplastic neoplasm or myelodysplastic myeloproliferative neoplasm, or one of these MDS-related genetic abnormalities, including a complex karyotype, which means more than 3 abnormalities in the karyotype, and 17p deletion and isochromosome 17q. These are p53 deletions. And we also look for mutations in these genes. And if it has one of these genetic abnormalities, or the patient has a prior history of an MDS or MDS-MPN, then it falls into AML myelodysplasia related, which has unfavorable prognosis. Now, if we've excluded all of these things, we finally get to AML defined by differentiation. And so most of our algorithm at this point has been based on genetics, but with AML defined by differentiation, we're giving it a phenotypic subtype based upon whether they have, for example, monocytic features, which would put it into acute monocytic leukemia, or let's say erythroid differentiation would put it into acute erythroid leukemia. And there's also basophilic leukemia, acute megakaryoblast leukemia, and so on. And so the WHO has a lot of very detailed rules around that. Remember how I mentioned that myeloid leukemia of Down syndrome and transient abnormal myelopoiesis of Down syndrome fall under a larger rubric of myeloid neoplasms associated with germline predisposition. And so there's a growing list of familial and congenital mutations that predispose to myeloid neoplasms, including Li-Fraumeni, Fanconi anemia, neurofibromatosis, etc. So when we classify these, we add these on as an attribute qualifier. For example, acute myeloid leukemia, myelodysplasia related, associated with germline TP53 variant. Myeloid neoplasms associated with germline predisposition are themselves under a larger category of myeloid neoplasms and proliferations associated with antecedent or predisposing conditions. And this larger category also includes myeloid neoplasm post-cytotoxic therapy. So the WHO has a very large list of cytotoxic therapies which predispose to myeloid neoplasms. For example, somebody who had breast cancer and got topo-2 inhibitors, or somebody who had lymphoma and got large fields of radiation, they are more predisposed to developing MDS, MDS-MPN, and AML, but with more unfavorable prognosis. And so this is also added on as a disease attribute qualifier onto the name of the larger original diagnosis. So for example, acute myeloid leukemia, myelodysplasia related post-cytotoxic therapy. And this category specifically refers to MDS, MDS-MPN, and AML, and not MPNs. The myeloid lymphoid neoplasms with tyrosine kinase fusions are fairly uncommon. They often show eosinophilia but have highly variable presentations. They can present like MPN, MDS, MDS-MPN, AML, MPAL, BALL, TALL. Some of them, especially PDGFR-A and PDGFR-B rearrangements, are highly responsive to tyrosine kinase inhibitors. So if you have one of these fusions, this category supersedes all other myeloid and lymphoid types, as well as systemic mastocytosis. So to recap, we've covered our major MPNs, the most common being CML and then ET, followed by PV and PMF. Then we covered our prototypical MDS-MPN, which is CMML, and very briefly touched on systemic mastocytosis. Then we went into a little more detail on MDS and AML. If we go back to our complete list of myeloid neoplasms, we covered almost all of the major categories, starting with myeloproliferative neoplasms, myelodysplastic myeloproliferative neoplasms, and myelodysplastic neoplasms. Very briefly in mastocytosis. Then we went into acute myeloid leukemias and myeloid neoplasms post-cytotoxic therapy, touched on germline predisposition and tyrosine kinase fusions. The two categories I didn't cover were clonal hematopoiesis of indeterminate potential and clonal cytopenia of uncertain significance, which are precursors to myelodysplastic neoplasms. And then acute leukemias of ambiguous lineage are leukemias that are either undifferentiated or differentiating in two different directions. Now on to B-cell things, in which I'm also including plasma cell neoplasms and Hodgkin lymphoma. Pictured here is a case of mantle cell lymphoma, where the classic description is medium-sized cells with very irregular nuclei, but forming overall a very monotonous proliferation. B-cell neoplasms are in this portion of the hematopoietic tree and account for about 60% of the incidence pie chart. The traditional way to organize B-cell neoplasms is by their presumed cell of origin, starting with the hematopoietic stem cell, which differentiates into a B lymphoblast, which undergoes VDJ recombination to become this naive B cell. Naive meaning that it hasn't seen antigen yet. This cell expresses IgM and, by alternative splicing, IgD, and this cell can circulate in the peripheral blood, or it can deposit in the mantle zone of a primary follicle, becoming a mantle cell. After stimulation by antigen, the circulating cell becomes activated to an immunoblast, And immunoblasts and antigen-activated mantle cells undergo somatic hypermutation, affinity maturation, and class switch recombination in germinal centers as centroblasts and centrocytes. Centroblasts and centrocytes are larger cells with high turnover, turning primary follicles into secondary follicles. Immunoblasts and germinal center cells can develop into long-lived memory B cells, and these cells can either circulate in the peripheral blood or deposit in the marginal zone, becoming marginal zone cells. Germinal center cells and memory B cells can further develop into long-lived plasma cells, which function as antibody factories. Diagnostically, we divide the life of the B cell into the immature stages and the mature stages. Immature B cell neoplasms tend to behave more aggressively than and are treated differently than mature B cell neoplasms. We further subdivide the mature stages into pre-germinal center, germinal center, and post-germinal center. We believe that these stages of development are the points of malignant transformation for these respective B cell neoplasms. B lymphoblastic leukemias are primarily found in the marrow because that's where their normal counterparts live. Mantle cell lymphomas appear to be primarily pre-germinal center, and chronic lymphocytic leukemias primarily circulate in the peripheral blood, but they're split between two stages because some of them seem to have a pre-germinal center DNA sequence pattern, but others seem to have a post-germinal center sequence pattern. Follicular lymphoma, Burkitt lymphoma, and Hodgkin lymphoma are germinal center derived, whereas diffuse large B-cell lymphoma is this kind of grab bag of 17 types in addition to diffuse large B-cell lymphoma not otherwise specified. So some of them are germinal center and some of them seem to be post-germinal center. Hairy cell leukemia seems to derive from a memory B-cell, marginal zone lymphoma from the marginal zone, and lymphoplasmacytic lymphoma and plasma cell myeloma are plasmacytically differentiated B-cell neoplasms. Categorizing B-cell neoplasms by their presumed stage of origin is useful conceptually, but operationally, it's not like the tumor calls you up and goes, I'm from Germinal Center City or I'm from the suburb of Mantle, right? Like, you have to figure it out. Classically, the major cut points are where there are large differences in therapeutic approach. And the first major cut point is, is it immature or is it mature? Because if it's immature, it's B lymphoblastic leukemia, which is an acute leukemia, meaning death occurs in weeks or even days if it's untreated. And typically, the way that we can tell that it's B lymphoblastic leukemia is that the marrow is chock full of these blast cells, which have very little or relatively little cytoplasm and this quote-unquote fine or very evenly dispersed chromatin. CD34 and TDT are pretty specific markers for immaturity, and the presence of CD10 or the lack of CD20 and the lack of surface light chain is usually supportive but not entirely specific. If it's mature, the next major cut point is it Hodgkin lymphoma or is it non-Hodgkin lymphoma? And this is because Hodgkin lymphoma has a greater than 90% cure rate. And I showed you a case near the beginning, but these will typically have a subset of these Reed-Sternberg or owl eye cells, and immunophenotypically they're very distinct from other B-cell neoplasms in that they're strongly CD30 positive and typically CD20 negative, and a subset of them will have CD15, which is typically a myeloid marker. If you're dealing with a non-Hodgkin lymphoma, the next major cut point is it small or is it large? Because large B-cell lymphomas tend to behave more aggressively and require treatment, whereas small B-cell lymphomas tend to behave more indolently and the treatment may be worse than the disease. Generally, they both have a standard B-cell phenotype, CD19 and 20 positive, with monoclonal surface immunoglobulin, but there are a good number of exceptions to that. And the way to distinguish them then is partly by genetics and partly with just 2 eyeballs and a brain. The last group that I'll cover are the plasma cell neoplasms, which are generally pretty distinct from the other B-cell neoplasms, and they're typically found in the bone marrow. Morphologically, plasma cells tend to have this very round eccentric nucleus with this basophilic cytoplasm and this perinuclear HOF or Golgi zone, which packages and secretes antibodies. Immunophenotypically, they're pretty distinct in that they tend to lack CD19 and CD20. CD138 is fairly specific. This super bright CD38 is a pretty good marker for them, as is VS38C, and they'll have monoclonal cytoplasmic but not surface immunoglobulin. With the small B-cell lymphomas, there are four main ones that we work up immunohistochemically before we get the genetics. The standard algorithm starts with CD5, and if it's CD5 positive, we look for BCL1. If it's BCL1 positive, that's strong evidence for mantle cell lymphoma. And a strong majority of these will have a T1114 BCL1 heavy chain rearrangement, which places the strong heavy chain enhancer near the BCL1 gene, which is also known as CCND1. This results in abnormal expression of the BCL1 protein, which is also known as cyclin D1. Some cases have alternative translocations which lack BCL1 expression, but these can typically be detected by SOX11 staining. Because mantle cell lymphoma is more aggressive among the small B-cell lymphomas, we often use a combination of BCL1 and SOX11 in order to increase our sensitivity. If you have a CD5 positive BCL1 negative small B-cell neoplasm, it's usually chronic lymphocytic leukemia. And unlike with other small B-cell neoplasms, CLL usually has dim CD20, dim surface immunoglobulin, and diminished CD79B. CD79B is the co-receptor for surface IG, so they usually go together. LEF1 in the context of a small B-cell neoplasm is fairly specific for CLL, but outside of that context, it's also positive in background T cells, and in the bone marrow, it's also positive in developing B cells or hematogones. We don't do LEF1 or BCL1 by flow cytometry, so in flow cytometry, we use CD23, diminished CD81, and CD200 to distinguish it from mantle cell lymphoma. If we're dealing with something that's CD5 negative, we then look at CD10. And CD10 in the context of lymph node is a marker for germinal center. Outside of the context of lymph node, it's also positive in hematogones and B and T lymphoblasts in the bone marrow. So if you have something that's CD10 positive and a mature small B-cell lymphoma, it's usually follicular lymphoma, which 90+% of the time has a T1418 Ig heavy chain BCL2 rearrangement, which places the heavy chain enhancer near the BCL2 gene and causes BCL2 to be abnormally expressed. BCL2 is an anti-apoptotic protein which keeps normal B cells alive when they're not stimulated by antigen. But in the germinal center, the B cells should be kept alive by antigen stimulation, not BCL2. So BCL2 should normally never be expressed alongside germinal center markers like CD10, BCL6, or HGAL. But BCL2 is not specific for follicular lymphoma. It's usually actually expressed in all of the small B-cell lymphomas as well. And unlike the other small B-cell lymphomas, follicular lymphoma almost always lacks CD43 and MNDA. A CD5 negative, CD10 negative phenotype on a small B-cell lymphoma favors marginal zone lymphoma. But follicular lymphoma can lose CD10, and mantle cell lymphoma and CLL can occasionally lose CD5, so you have to be careful. In the context of a CD5 negative, CD10 negative lymphoma, CD43 is supportive of MZL, but it's not sensitive. MNDA is fairly sensitive and specific in this context, but it's also present on a subset of mantle cell lymphomas and CLLs. These are the important genetic alterations to know in B-cell lymphomas. I barely mentioned Burkitt lymphoma, but it's an aggressive germinal center-derived lymphoma, which most commonly has a T814 rearrangement, which places the Ig heavy chain enhancer near the very powerful c-Myc oncogene. Less common translocations are to the Ig light chains on chromosomes 2 and 22. c-Myc translocations are required for a diagnosis of Burkitt lymphoma, but they're also seen in large B-cell lymphoma. I've already talked about follicular lymphoma and mantle cell lymphoma. Lymphoplasmacytic lymphoma is a post-germinal center small B-cell plasmacytoid neoplasm, characteristically with the MIDI88 L265P mutation. A minority of cases have a CXCR4 S338 nonsense or frameshift mutation. In some cases have both of these simultaneously. Hairy cell leukemia is an indolent small B-cell lymphoma, which is typically found circulating in the blood with quote-unquote hairy morphology. These have a BRAF V600E mutation, which is the same as in papillary thyroid carcinoma, melanoma, and Langerhans cell histiocytosis. With B lymphoblastic leukemias, the favorable alterations include the T1221 ETV6 RUNX1 rearrangement and hyperdiploidy. This RUNX1 is the same RUNX1 on chromosome 21 as seen in favorable prognosis AML with T821 RUNX1 RUNX1T1. With hyperdiploidy, blasts should contain greater than 50 chromosomes, but usually have greater than 66. The unfavorable alterations to know include the T922 BCR-ABL1, which is often written in patient charts as Philadelphia positive or PH positive. Rearrangements with 11q23 KMT2A happen with a large number of partners and are also of unfavorable prognosis. And with hypodiploidy, the technical definition is less than 46 chromosomes, but unfavorable prognosis is really only associated with 43 and below. With CLL and plasma cell myelomas, TP53 mutation or loss is unfavorable, and with large B-cell lymphomas, a c-Myc rearrangement simultaneous with a BCL2 rearrangement specifies an aggressive subtype of DLBCL/high-grade B-cell lymphoma with MYC and BCL2 rearrangements. So going back to our giant incidence pie chart, I've briefly overviewed with you B lymphoblastic leukemia, Hodgkin lymphoma, Burkitt, LPL, mantle, marginal, follicular, CLL, large B-cell lymphomas, and plasma cell myelomas. If we go back to our giant list of diagnoses, I've briefly overviewed B lymphoblastic leukemias. Small B-cell lymphomas have a lot of different subtypes, and they can also transform into large B-cell lymphomas, a category called transformations of indolent B-cell lymphomas. Although with mantle cell lymphoma, we tend to refer to the ugly ones as blastic or pleomorphic rather than DLBCL. And there are 18 total types of large B-cell lymphomas, including the not otherwise specified type. We touched on Burkitt lymphoma, didn't really talk about KSHV or HHV8. We briefly touched on Hodgkin lymphomas and plasma cell neoplasms. The WHO 5th edition now includes these non-malignant or benign-ish things that are pretty grab bag, and I didn't really cover them, but they're called tumor-like lesions with B-cell predominance. Now on to T-cell things. This is a nice case of what used to be called angioimmunoblastic T-cell lymphoma and is now called nodal T follicular helper cell lymphoma angioimmunoblastic type. And what you see is a proliferation of small to medium-sized lymphocytes with some cleared cytoplasm and surrounding fibrosis in a background of increased vasculature. T-cell neoplasms are this portion of the hematopoietic tree and 5% of the incidence pie chart. The life of the T-cell starts in the bone marrow with a hematopoietic stem cell, not pictured here, which can differentiate into a progenitor T-cell or prothymocyte. These can go out to become NK cells, which travel to spleen or mucosa or circulate in the peripheral blood. The prothymocytes can also travel to the thymus to become subcapsular cortical thymocytes, which can differentiate into gamma delta T cells, which can travel to spleen or mucosa or circulate in the peripheral blood, and can also go to the skin. These subcapsular cortical thymocytes can further differentiate into common thymocytes, which express both CD4 and CD8, as well as TCR alpha beta. These can then either lose CD8 to become CD4 positive medullary thymocytes, or they can lose CD4 to become CD8 positive medullary thymocytes. These can then circulate as CD4 or CD8 positive naive T cells, which can be activated by antigen to become CD4 or CD8 positive T immunoblasts. The CD8 positive immunoblasts can differentiate into memory or effector cells, while the CD4 positive immunoblasts can differentiate into Th1, Th2, Th17 cells, follicular helper T cells, or T regulatory cells. As with the B-cell neoplasms, we believe that the various T-cell neoplasms originate from specific stages of maturation. Extranodal NK T-cell lymphoma is an aggressive EBV-positive lymphoma often found in the nasal cavity with both T and NK cell differentiation, hence NKT, so it expresses both the T-cell marker CD3 as well as the NK cell marker CD56. Hepatosplenic T-cell lymphoma is an aggressive T-cell lymphoma which usually expresses TCR gamma delta, as well as having a typical gamma delta T-cell phenotype, CD3 positive, CD5, CD4, and CD8 negative. T lymphoblastic leukemia is an acute leukemia with a developing T-cell phenotype, often expressing the immature markers CD34, TDT, and or CD1A. CD10 is helpful, and some of them co-express CD4 and CD8 like common thymocytes. T prolymphocytic leukemia is an odd one. We don't really know where it comes from. It seems to have post-thymic genetics, but it also has immature features like some of them can co-express CD4 and CD8 with some dim CD3. Regardless, they're usually aggressive circulating neoplasms with very high white counts, which express TCL1 and bright CD52. The most common T-cell neoplasms are mature CD4 positive lymphomas, and the one that we see most commonly is mycosis fungoides and its circulating counterpart, Sézary syndrome. On the flow service, we're looking for CD4 positive T cells with loss of CD7 and or CD26, and commonly they have slight fluorescence shifts in CD3 and CD4. Peripheral T-cell lymphoma, not otherwise specified, is kind of a wastebasket category where we throw in any CD4 positive T-cell lymphoma that doesn't fit in anywhere else. But in general, it has a fairly poor prognosis. Anaplastic large cell lymphoma, I think of as the T-cell version of Hodgkin lymphoma, in that in children it's potentially curable, and like classic Hodgkin lymphoma, it expresses CD30 and often lacks markers for its lineage, especially CD3. It also frequently co-expresses myeloid markers like CD13 and CD33. Notable T follicular helper cell lymphomas are CD4 positive lymphomas, which come from the follicular helper T cells of germinal centers. So they have some of the same markers as germinal center B cells, namely CD10 and BCL6. And they also express PD-1, which is important in many immunotherapies. Adult T-cell leukemia lymphoma, or ATLL, is a typically CD4 positive T-cell lymphoma resulting from transformation by human T lymphotrophic virus 1, or HTLV1, which is endemic in Asia. It's a lymphoma of T regulatory cells, and so it expresses CD25 and FOXP3. The CD8 positive T cell lymphomas are much less frequent than the CD4 positive ones. With SPTCL and EDL, you can kind of just know that they're CD8 positive, but they're pretty rare. The one that we see most commonly is probably T cell large granular lymphocytic leukemia, or TLGL leukemia, which is typically an indolent disease with an increase in circulating large granular lymphocytes associated with neutropenia. And they usually express CD3, CD8, CD57, and CD16. There are a handful of T-cell lymphoma mutations that are probably the most characteristic. You see the T25 ALK NPM1 rearrangement in ALK-positive anaplastic large cell lymphoma, which has much more favorable prognosis than ALK-negative anaplastic large cell lymphoma. It's not entirely clear that DUSP22 rearrangements are really favorable in ALK-negative ALCL, but TP63 rearrangements are unfavorable. 90% of TPLLs have an inversion 14 or T1414 rearrangement, which explains the strong expression of TCL1. Most hepatosplenic T-cell lymphomas have an isochromosome 7q, and adult T-cell leukemia lymphomas have integration of HTLV1. Interestingly, we think of TET2 and DNMT3 mutations as more myeloid, but you also see them in angioimmunoblastic NTFHL. And common to several of these are STAT mutations as well. Lastly, we'll do a tiny bit on lymphoid proliferations and lymphomas associated with immune deficiency and dysregulation, or IDD. These used to be scattered across several chapters in the WHO, but are now unified under a single framework, and they have to be reported in 3 parts. The first part is the histologic diagnosis, whether it's hyperplasia, polymorphic LPD, mucocutaneous ulcer, or lymphoma, which you would classify similarly as you would for an immunocompetent patient. The second part you have to report is the viral association, whether it's EBV, KSHV, HHV8, or a combination of them. And the third thing is the immune deficiency or dysregulation setting. Inborn errors of immunity are primarily immunodeficiencies, for example, DeGeorge syndrome, SCID, CVID, Wiskott-Aldrich, ataxia telangiectasia, XLP, ALPS. There are hundreds of them. Other settings include HIV infection, post-transplant, autoimmune disease, iatrogenic or therapy-related, including chemotherapy for other diseases, CAR T and checkpoint therapies. And then there's also this very vague thing of immune senescence, which at least in part might be meant to capture older individuals in general. And that's it.